and cartilage, you call this a bronchus. From there, what is the next part of the bronchial tree, the next name?
Bronchiole.
The bronchiole. And what is typical for a bronchiole? What do you remember?
It had a different epithelium.
Yes, in which way? No cartilage anymore, exactly. So it loses cartilage. So if you have, you see here are big ducts here with cartilage in the wall here, and then here's a little one which doesn't have it anymore, or here's one, or here's one. It's still quite big, but you don't see any cartilage. Okay, and that's probably a bronchiole, or maybe this is a smaller bronchiole here, but this is even larger and does not have any cartilage. So we can zoom in. Let's see. Yeah, so there's no cartilage left here. What else do you know? So no cartilage. What is the next thing you should know about the bronchiole? So the bronchiole usually does not have, per definition, does not have glands anymore. This is unfortunately not really true here. Look at this. Can you see these glands here? So there are still some packages of glands. So probably this part here of the bronchial tree is somewhat in between what we call a typical bronchus and a typical bronchiole. So the cartilage has stopped. There is still a few glands, but those will disappear pretty soon when we go further down that bronchial tree. You see here in this particular part, which is right in the neighborhood to this here, there is actually no gland in the wall anymore. So this part here is really typical, like a bronchiole should be, and this is something in between. Okay, so let's focus on this part, and you can see now there is no gland anymore here in the wall, and there is no cartilage, and there is an epithelium which now is more and more just a simple epithelium. It was a pseudostratified respiratory epithelium, and a bronchus has usually a very typical respiratory epithelium. But now, when it branches more and more and it becomes bronchioles, the epithelium will change from a pseudostratified one to a simple one, which then will become more and more flatter. So at the beginning, it's still kind of columnar, and then it will be for quite some time just cuboidal. And here, when you look at this, it seems to be quite cuboidal, because you can see here there's one row of cells and they have round nuclei, and it might be still a little bit columnar, but it really tends to become cuboidal more and more. And you should have less and less goblet cells, and you should have less and less ciliated cells. There are some, and in the beginning there are probably still a lot, but the farther you go within the bronchial duct system, a bronchiole duct system, the more these cells disappear. But there is a new cell type which appears instead. Who remembers what that cell type was?
Club cells?
The club cells, exactly. So what is the job of club cells?
It produces surfactant proteins, and so helps prevent collapsing of these
Yes,
airways.
exactly. So some of the molecules which are part of the surfactant are produced by these club cells. And then there is another job that the club cells have. They produce, yeah.
I don't know, maybe reduce capillary pressure.
No,
Okay.
they produce things which help to kill bacteria. So they have antimicrobial microbial molecules that they can secrete, and they are part of the innate immune system. Okay, so unfortunately, in this particular specimen, it's hard to identify the club cells because I don't know, the fixation also did not work well here. But there's one more specimen that I want to show you where you can actually see them quite nicely.
Professor, did you
Yes.
say they produce antibacterial substances?
Yes.
Okay.
And what do you think is this here? Or here's the same. And here. Here's a little bit. But here and here it's here it's easier to identify it. How does it look to you?
Endothelial cells. No.
Then you need a lumen.
Right.
So this is the big lumen of the bronchiole. Here is the epithelium. As we just discussed, it becomes now more cuboidal, simple. Goblet cells and ciliated cells will decrease in number more and more, and club cells appear instead. But one feature of a typical bronchiole has not been named yet, and you can see it here. Bronchioles have these cells here. When we go back to our bronchus, now look at the bronchus here. And when we now go back, look at this. And when we now go back, here it is again, but not in such a, it's not a continuous layer anymore.
Is it alveoli? Or?
No, the alveoli are these
big
spaces here. These are the alveoli. But this here is smooth muscle tissue. So you still have in the bronchioles smooth muscle tissue. Okay, so the bronchioles can contract and relax like the bronchi, not as much. As you can see, there's less smooth muscle tissue, sometimes not even a continuous layer anymore, but still you can make the lumen here a little bit smaller. Okay, so when we repeat this, a typical bronchiole has a cuboidal simple epithelium, less and less ciliated cells and goblet cells, but instead club cells appear. And then there is smooth muscle tissue also. And it's like the one in the bronchi. It's innervated by the sympathetic nervous system, which initiates or triggers relaxation. So under stress, the bronchioles are also wider. Okay, so these are typical features of bronchioles. This, by the way, these are blood vessels, but they are not normal. You can see they have a very, very thick media, and this is a pathological feature here. This is not normal. So you should not focus too much on these guys here because they don't look good. So you should ignore those. Okay. Yeah. Yeah. Shoot. Yes. What the pathological conditions were? I've no idea. This is from an animal, as far as I remember. Um, Porcine or feline, it says here. And I have no idea why this animal had these very, very thick vessels. I have no idea where this comes from. So that's quite strange. But for some reason, this animal had it. I have no idea where it comes from. I'm sorry. Yeah. But a normal blood vessel would not have such a thick media here, because you see how small this lumen here is. And then you have this really, really very, very thick media. This is really not okay. Like here, this one too. And here, so that's so focus just on the bronchi and the bronchioles. They look normal and quite good. And then you can see now here everywhere the bronchioles. So all these empty space alveoli, all these empty spaces now are alveoli. And you can imagine that our bronchi, they branch and become smaller and smaller, and then the branches turn into bronchioles, and the bronchioles branch and become smaller and smaller. Here you can see there's even a smaller one here. And you can see now a very nice cuboidal simple epithelium lining everything here now. And look at this here. Now you can even see the club cells nicely. Here, look at this. Can you see how they bulge forward, which is a typical feature? That's why they have this name, club cells. Here is one, here's another one, here's one, here's one, here's us a few. And they bulge forward a lot, and that's why they are easy to be identified. Okay, so this is also a nice, more kind of terminal bronchiole now. Here's still some smooth muscle cells. Okay, you can see the elongated nucleus and the elongated cells here. This is smooth muscle tissue. And here's the typical epithelium. It is contracted somehow so that the epithelium runs in these waves here and these curves. That comes from the shrinking mostly. Okay, when the tissue shrinks during fixation, not always you have these big folds in the epithelium. When you have a normal lung filled with air, it would not look like this. It would be more open. Okay, but here you have a lot of folds in the epithelium now from shrinking and from that fixation and so on. But here you actually can see a very nice typical more distal bronchiole with a nice cuboidal epithelium and with club cells. I think here now everyone can get the idea why they have their name. And you still have some smooth muscle tissue, which is also a typical feature of a bronchiole. So these orange dots here actually are red blood cells. Okay, they turned kind of orange here in this specimen for some reason, but they are red blood cells. So you get an idea already that even in these very small interalveolar septa, and that is how these walls are named now, in between neighboring alveoli, you have these walls and they are called a septum. And if you have multiple, you have septa, that's the plural form of septum. And these septa are named interalveolar septa because they are in between these two air-filled spaces. And when you look at such an interalveolar septum, you can see that red blood cells are even within this very tiny wall. And you all know that these walls contain little capillaries, which are part now of the barrier between the air and the blood. So there's a very thin wall, which now is the only barrier between blood and air, so that when we inhale, we can take up oxygen easily, and when we exhale, so that we get rid of the CO2. For that reason, we need a very thin barrier, and that is here in that wall. And the next specimen shows this much nicer. That's why we should not spend too much time on these interalveolar septa, because the other specimen is much better. It has a better resolution, a better fixation, and so on. But this specimen is very good to look at late bronchioles, early bronchioles, where you even have a few glands, and the bronchi. Okay, any questions related to this specimen here? If that is not the case, we switch to 150, which is also a lung tissue. And here it says this is a lung from a rat. The macrophages have stored some coal particles again, so there are some inert particles, black particles, which could not be broken down by the cells, so they have accumulated all this. And you can see now where the cells are that actually phagocytize everything that manages to get all the way to the alveoli. And this is a semi-thin section, so this specimen was probably fixed with a perfusion fixation and then embedded in araldite and cut, and the staining is methylene blue. So as you can see, most things are blue here in this staining. And here you can see details of the different cell types which we have in the bronchioles and even in the interalveolar septa. So when we go one more time here into this larger lumen, which probably is some bronchiole, some late bronchiole, then you can see the club cells here bulging forward. Look at this. You have a simple epithelium, and here are the club cells bulging forward, and here another one, here another one, here and here. So there are now these club cells. So this is a bronchiole with a cuboidal epithelium. Here is a big blood vessel with lots of red blood cells. They are stained blue now here in this methylene blue technique, but here you can see when we zoom in, you can see nicely now the red blood cells. You can see, look at here at the bottom, there is an endothelial cell with the nucleus bulging forward a little bit. So this is the very thin, here's another cell body of an endothelial cell. Here's another one here. Here's a nice one, here's a nice one. Here are two. So you can see the endothelium. You can actually see material of the internal elastic membrane. Look at this, these blue dots, the dark line here. This is the internal elastic membrane, and then you have a very thin layer of smooth muscle cells, but they are hard to identify. I only know that they must be there, so it's hard to identify them. But when you look closely, you can see the endothelium here. You can see material of the internal elastic membrane. So it's possible. Okay, so here's a nice blood vessel. And when we go further to the side, we now see all these alveoli. We can see the interalveolar septa. And when we know now how red blood cells look like, and we go into these walls here, you can see here are red blood cells in a capillary. Here's a capillary with red blood cells. Here's a bigger one. Here's a very small one. And here you can see a red blood cell. Okay. Here's a little capillary, and here you can see a red blood cell. Here's a capillary, and here's a red blood cell. Okay. So you need to get used to how to look at these specimens so that you are able to catch the patterns of specific things that you want to see. So my question for you guys now is, which cell types are we looking for now when we are at the level of the alveoli and we look at these interalveolar septa? What are we looking for? What do you want to see? What are the typical cell types?
Pneumocytes 1 and 2.
Pneumocytes 1 and 2, or alveolar cells, type 1 and type 2? Exactly. Both names are correct. So how does a type 1 cell look like, and how does a type 2 cell look like? Let's start with the type 2 cells. A type 2 pneumocyte or type 2 alveolar cell. What does it do? What's its job, and how does it look like?
Sorry, I don't know how it looks like. All I know, it makes surfactant.
It's the major cell type to produce surfactant. That's correct.
So it has to have a lot of glands, perhaps?
No, a cell cannot have glands. A cell is a cell.
Okay.
A goblet cell is a gland because it's a one cell gland, but a cell cannot have glands. It's not the right way to put it.
Okay, I meant to say goblets also, perhaps.
Yeah. No, but I mean, when a type 2 cell is producing something in high amounts, you were probably thinking of something right, but you just put it the wrong way. So what might type 2 cells have in order to produce surfactant? Something which you can see in a histological section. Maybe it has some storage vesicles. You know, it produces something, stores it, and then it releases these things once in a while. Okay, so the type 2 cells are actually cuboidal cells. They are not squamous like the type 1 cells. Type 1 cells are completely squamous. You can see one here. Look at this. There's one cell which bulges forward here, and the rest of the cell body is very, very thin. So everything here is lined by very thin and squamous type 1 cells, and once in a while you can see the nucleus, and there the cell body is a little bit thicker and it bulges forward just like endothelial cells in the blood vessels. Okay, so when you see a cell body bulging forward to the airway side here, then it's a type 1 cell. The type 2 cells are these guys here, which are quite big and bulging forward, and you can see one here. You can see another one here. You can see another one here. So regularly, look at this, you have these big cells here, another one, bulging forward into the alveoli, and they are quite big and easy to identify for that reason. And here you can see they have these, storage vesicles. These, you have, it looks like lots of holes. And as you know, they produce the surfactant, and surfactant has multiple components, lots of lipids, for example. These cells produce phospholipids, which are comparable to those which we have in the cell membrane. Each cell membrane has 2 layers as you know, of phospholipids, and the phospho part shows to the outside and the lipid component shows to the inside, so that the two lipid components meet in the middle of the cell membrane. And if you only take half of that, just one phospholipid, you can create multiple layers with this kind of thing, not just two. You can kind of create multiple layers, and this is what is done in the surfactant. And the type 2 cells, they have these Oatland bodies, they are called, after a guy who described this. Some of these vesicle-like structures that we see here are full of phospholipids, which are secreted by the type 2 cells and which are an important part of the surfactant. So you can identify the type 2 cells pretty easily. They are big, they are bulging forward, they are not squamous, and they have these storage vesicles, which you can see even in a normal light microscope quite easily. Okay. And one major job is to produce surfactant, and the other major job, you have not named that yet. What are type 2 cells also doing?
They are progenitor cells. So
exactly.
they differentiate to the type 1 cells.
They are the progenitor cells, and they can proliferate once in a while, and a daughter cell can develop into a type 1 cell. So they help to replace the epithelium regularly, which now lines the surface of these alveoli. So type 2 cells, and here you can see two, and here another one, and type 1 cells, which are so squamous that you hardly can see them. But when you start looking, you should once in a while see cell bodies bulging forward a little bit. Here maybe just, it's very tiny. Here maybe there's one. That's probably one. Okay, so these are the type 1 cells, and they are very, very flat, and they line the surface of these alveoli. Okay, and then of course you have the capillaries inside of these walls, these interalveolar septa, and they are normal capillaries with endothelial cells. Inside are red blood cells. And so if you want to define how the barrier looks like between the air and the blood in the capillaries, which different layers do you have in that barrier? Who wants to try to remember what the barrier looks like? So the gas, oxygen or CO2, has to go through which layers, which are all very thin, but you still have them? Who wants to give it a try?
The endothelial layer.
So the endothelium on the blood side, you have
Blood
the endothelium, okay, and the endothelial cells, they sit on usually they sit on whatever. What are they attached to?
The basal cells.
Not basal cells.
Basal lamina.
The basal lamina, exactly.
Yes.
So they have a basal lamina, and what is the next layer?
Then the alveolar layer, alveolar cells.
Exactly. So then the type 1 alveolar cells, they also attach to that basal lamina, but from the other side. And then the type 1 cells are covered by surfactant. So it's the endothelium, the basal lamina, the type 1 cells, and surfactant. These are the 4 layers that the gases need to go through, but it's easy for them. Okay, so it's very thin, as you can imagine. When you look at this here, sorry, wait a minute. You can see here's the lumen of the blood, the capillary, and then you can see how thin the wall is. And this wall contains all these 4 layers that we were talking about. And here on the other side, the same. Okay, and here's another type 2 cell. And whoops, what is this here? This big guy?
Isn't that our macrophage with colon in?
This is an alveolar macrophage. Exactly. You can see the nucleus here in the center of the cell. And then you can see all these dots here. These are phagosomes with coal particles. Okay. So the cytoplasm is full of phagosomes with these coal particles. So that would be one big macrophage. Here's another one. And here's another one. Okay. So you can see how many macrophages we have here, some smaller ones, but still the same. Okay, you can see the nucleus of that macrophage here, nicely visible. So you get an idea how many macrophages we have in our lung, and that is normal. Okay. Okay. So any questions related to this specimen here? So we have a nice bronchiole here with club cells, and then we have these nice interalveolar septa with blood vessels, the capillaries. We have type 1 and type 2 pneumocytes, and we have macrophages. There are still some more cells, some fibroblasts and so, but they are hard to find. So we probably should not spend too much time for that. Any questions related to what we talked about right now? Not the case? Okay, then I suggest we switch to another topic, the kidney, 117. I have here 117, and there's 2 options, A and B, and I suggest we take B